Welcome to another episode of The Sleepy Space. Close your eyes now. Let your breath slow down like ripples fading on a still pond. Within you, past your bones and blood, past thoughts and skin, there lies something quiet, something smaller than you've ever imagined. You cannot feel it move, but it moves. You cannot hear it speak, but it sings. In every cell of your body, trillions of them, there lives a thread, a spiral staircase of code. That thread is DNA. It's curled tightly like a whispered secret, hidden in the heart of nearly every cell. DNA is not alive by itself, but it holds the instructions for life. It is not conscious, but it carries a memory of the past so ancient it predates your name, your story, even the shape of your hands. Imagine for a moment that your body is a great library. Each cell is a room, softly lit, lined with bookshelves. On every shelf rests a scroll, and inside that scroll the same long, elegant script. This script is written in 4 letters, A, T, C, G, and they repeat again and again in careful patterns, not in English, not in sound, but in a molecular rhythm that speaks to the cell like a lullaby. A for adenine, T for thymine, C for cytosine, G for guanine. These are not just chemicals, they are the alphabet of you. DNA isn't just information, it's a map and a memory and a mission. It tells your cells how to grow, when to divide, what to become. It decides the color of your eyes, the shape of your heartbeat, the way your hair curls when it rains. But even more than that, DNA carries time. Some of the letters written into your genes have been passed down through thousands of generations. Your ancestors, people who lived and laughed and wondered long before you, left traces inside you, tiny echoes folded into the double helix. The double helix, that's the shape DNA takes. Imagine a twisted ladder spinning softly upward in a spiral. Each step of the ladder is made of 2 letters joined together. A pairs with T, C pairs with G. These pairings are like lovers dancing, locked in perfect balance. The shape holds strong, yet flows with flexibility, like silk in the wind. And yet, as vast and mysterious as it sounds, this entire code fits into something unimaginably small. If you stretched out the DNA from just one of your cells, it would be about 6 feet long. Now imagine folding that 6 feet into a space invisible to the naked eye. That's what your body does over and over again, packing entire novels into microscopic worlds. It's not just one copy, it's not just 2. Every one of your trillions of cells has its own copy of your DNA, like each cell is holding its own edition of your story. And each morning, as you wake, stretch, and breathe, these silent books are read. Your cells are the readers, your DNA, the text. But not every page is read at once. Cells read only the chapters they need. A cell in their eye might turn to the page about light. A cell in your liver may open to the part that speaks of detox and repair. The rest of the book remains closed, quiet, waiting. This selective reading is part of what makes you complex. All your cells carry the same book, but they do not become the same. Some become skin, others muscle, others blood or bone or brain. They are united by the same story, but they interpret it differently, like readers seeing poetry in different lines. DNA also replicates. It copies itself with astonishing accuracy. Imagine copying a 3 billion letter-long manuscript by hand over and over again every time a cell divides. That's what your body does, not with pens, but with proteins. Proteins called polymerases zip along the DNA, reading and building, like tiny librarians retyping ancient scrolls. Of course, sometimes there are typos. Tiny mistakes can slip in. Letters swapped, skipped, or repeated. Sometimes they're harmless. Sometimes they create change. These changes are called mutations. Not the kind of mutation you see in superhero stories. These are gentler, subtler shifts. They may change the way a cell works. They may cause illness or spark evolution. Because every so often, a tiny change opens a new path in the forest of life. You are, in part, made of mutations. You are the result of millions of years of trial and error, of scripts edited not by pens, but by nature itself. And now we can read that script. Only a few decades ago, reading the full sequence of a single human genome was an immense feat, requiring years and billions of dollars. Today, it can be done in hours. Your body's instruction manual, once locked in mystery, is now being slowly translated. And with that translation, a new possibility arises. If we can read this book of life, could we one day edit it? This is where our journey begins to drift forward, toward something unexpected. Because deep inside bacteria, scientists uncovered something curious. A system nature invented to defend against invaders. Something ancient, something precise. At first glance, it seemed small, but beneath its simplicity, it held a key. A key that could change how we understand disease, how we understand inherited traits, how we may someday write healing into the pages of our own biology. But not just yet. For now we are still here with you, resting in the quiet beauty of your body's design. Your cells, your strands of DNA, your unique and ancient code, all of it humming in gentle rhythm. It's a hum you don't hear, but it is always there. You are alive because that song continues, unbroken, unreadable to your ears, but understood by every cell. You are, quite literally, made of story. So tonight, as you drift deeper into rest, know this: you are the keeper of something remarkable. A language written in lightless rooms, in silent spirals across time. And somewhere, in the smallest corners of your body, the next page is already turning. The question lingers like mist in morning light. If we can read this story, what might it mean to edit it?
You're still resting, your body sinking deeper into quiet. Your breath has slowed. Your thoughts move like gentle waves now, not chasing, just floating. And as you rest here, we travel again, not outward, but inward, to a place smaller than a dust mote, smaller than a cell. This time, we meet one of the oldest forms of life on Earth, the bacteria. Bacteria are everywhere, in your skin, your gut, the air you breathe. They've lived for billions of years, long before humans wrote books or built cities. You might think of them as simple, but they are clever. Not clever like a scientist with a chalkboard, but clever like a seed finding sunlight, like a whisper that knows how to protect itself. Bacteria have enemies too, tiny predators called viruses, specifically bacteriophages or phages. Phages are so small they make bacteria seem enormous, and all they want is to inject their own DNA into a bacterial cell, hijack its machinery, and take over. They do this with precision, like a lockpick sliding into a keyhole. But bacteria are not defenseless. They've learned, slowly, over eons, to fight back. And one of the most beautiful ways they do this is through something called CRISPR. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, sounds complicated, but its soul is simple: memory. It's the bacterial immune system, made not of white blood cells, but of code. When a virus attacks, the bacteria fight back by saving a small piece of the viral DNA, like a photograph or a fingerprint. They store it in their own genetic code, tucked away in a special place. This memory stays with them, like a note passed from one generation to the next. So if the virus returns, the bacteria recognize it instantly, and they send out a special helper, Cas9, a protein shaped like a pair of scissors. Cas9 moves through the bacterial cell like a silent guard. It carries the memory of the virus, checking every strand of DNA it encounters. When it finds a match, it cuts, clean and precise. Snip. The invader is silenced. The threat dissolves into fragments. This is not rage. This is not violence. This is a kind of peacekeeping, written in molecules. The bacteria don't just fight, they remember, they record, they edit.
Let yourself drift a little deeper now. Your breathing is soft and slow like moonlight over still water. You are resting, not just your body, but your curiosity, letting it stretch out, wander gently. In this quiet, you return to the book of life, the pages made of DNA, the language you carry in every cell. But tonight, something is different. Tonight, we don't just read the script. We explore how slowly and carefully we are learning to revise it. Imagine a single sentence. The child will see the sunrise. Now imagine changing just one letter. The S becomes a D. Suddenly it reads, the child will die the sunrise. A simple slip and everything changes. This is what happens in genetic disorders. Just one letter in your DNA, one base pair out of 3 billion, can shift the instructions your cells follow, like a wrong note in a lullaby. Some of these errors cause little harm. Others can lead to diseases passed down from parent to child, etched into the code like a ghost in the margins. But now with CRISPR, we hold a gentle pen, a tool to erase the mistake, to change the D back to an S and restore the meaning of the story. One of the first successes came with sickle cell disease, a condition that distorts red blood cells into crescent moons, causing pain, fatigue, and shortened lives. Using CRISPR, scientists were able to reach into the bone marrow where blood begins and edit the DNA, reminding the cells how to make round, healthy ones again. It wasn't a miracle, it was science, quiet, methodical, but filled with hope. For the first time, a disease rooted in a single litter was softened by the hands of those who understood the language of life. Imagine the comfort of that. To know that a child born with a burden might one day be free from it, not by hiding the pain, but by correcting the page it was written on. And this is only the beginning. Scientists are now exploring how CRISPR might help in other conditions: muscular dystrophy, cystic fibrosis, some forms of blindness. Each one a different kind of misprint in the genetic text. Each one a puzzle of timing, delivery, precision. But the map is growing clearer. CRISPR can also make cells more resilient. In a clinical trial, researchers used CRISPR to edit immune cells so they could better recognize and fight cancer, not by destroying the body, but by strengthening its natural defenders. These cells were taught a new chapter: how to see danger more clearly and respond with strength. Even viruses, those ancient foes like HIV, are being challenged by CRISPR. By editing the DNA where viruses hide inside human cells, scientists hope to cut out the infection like trimming a vine from a tree. But it's not all healing and promise. This power is delicate. It must be used with care, with humility. Because editing DNA is not just changing a sentence. It's changing a sentence that will echo across generations. In 2018, the world was shaken by the birth of two little girls, twin sisters in China, born from embryos edited with CRISPR. Their DNA had been altered not to cure a disease, but to resist HIV, a virus their father carried. The news was not met with celebration, it was met with concern. Because the changes made would not just affect them, but all their children and their children's children. The edits were permanent and passed on. The scientific community had not agreed on this. There had been no consensus, no full understanding of the risks. And so the story darkened, not because of the tool, but the haste. CRISPR had shown us what could be done, but now we had to ask what should be done. This is the delicate place we now rest in. CRISPR is like fire in the hands of early humans. Warmth, light, survival, yes, but also danger if left uncontrolled. To edit the script of self is to walk a line between compassion and caution, between possibility and responsibility. So scientists move slowly, each discovery tested, each edit watched carefully, because this is not fiction, this is your body, your inheritance, your future. And yet, in the quiet of the lab and the hush of late-night research, more is being learned. Like how CRISPR can be used not just to cut, but to rewrite, to change a single letter without breaking the strand. This gentler version is called base editing. Instead of scissors, it uses chemistry, a brush instead of a blade. Imagine standing in a library with a pen that can change one letter in a book without tearing the page, without even touching the rest of the line. That's what base editing offers. It is slow, precise, and remarkably elegant. This precision opens doors, not just for treatment, but for prevention. Someday, parents may be offered choices to fix a gene before birth, to prevent a child from inheriting a life-threatening illness. And here the questions become heavier, more tender. What is health? What is normal? Where does healing end and enhancement begin? Can we choose the color of a child's eyes, their height, their intelligence? Should we? These aren't just scientific questions. They are human ones. They touch the soul of what it means to be alive, to be imperfect, to be shaped by something older than choice. And yet the editing continues, not recklessly, but with hope. Because most of CRISPR's early power is not in changing the unborn, but in helping those already here, the sick, the suffering, the waiting. You, lying here in the dark, are part of a species learning to write again, not with ink, but with enzymes, not with dreams, but with data, guided by something as old as instinct, the desire to understand and heal. Tonight you've traveled through the script of self. You've seen how letters can be changed, how pages can be corrected, how the story might continue more gently. But now another chapter waits. Because for all this power, for all this potential, there is something else that follows every step of discovery. A question that cannot be edited out. What does it mean to change the code of life when life itself is built not only of genes, but of values?
You are still resting. The night has deepened. The air feels softer, as though it's folding around you. And inside this stillness, another kind of space opens. Not in the body or in the cell, but in the mind, where questions grow like quiet trees. CRISPR has shown us what's possible. We've seen how a single molecule, small and silent, can shift the course of illness, reshape a future, even rewrite the story of life. But tonight we slow even further. We pause in that space between doing and deciding. Because science, like fire, can warm or burn. It is not the flame that chooses, it is the hand that holds it. So we turn to ethics, the calm, careful voice that asks, just because we can, should we? It's not always an easy question. Ethics rarely shouts. It does not speak in formulas or headlines. It murmurs, often unnoticed, like wind through tall grass. But its questions matter, perhaps more than any answer. Picture this. A child is born with a painful genetic disease. CRISPR offers a way to remove that pain before it begins, to edit the gene, erase the suffering, give that child a life with less fear. It sounds simple. It feels right. Who wouldn't want to help? But then, imagine something more subtle. A family chooses to edit out a condition not because it's painful, but because it's different. They want a taller child, or a quieter child, or one with perfect eyesight, or perfect skin, or the same musical talent as their great-grandfather. Here the lines begin to blur. Where does treatment end and enhancement begin? Is a body still yours if its story was chosen before you could speak? The worry is not about science losing control, but about humans forgetting what it means to be whole. Perfection is not peace. Control is not compassion. And so in labs and lecture halls, in boardrooms and bedrooms, scientists, ethicists, parents and policymakers gather to ask the hard questions. Some ask, should we allow edits only when there's clear medical benefit? Others ask, who decides what's a benefit and what's a preference? Some say we must prevent suffering. Others say we must protect diversity, the very imperfections that make us human. Because when we talk about editing the genome, we're not just talking about molecules. We're talking about identity, community, equity. We're talking about who gets to choose, who gets left behind, and what kind of world we are gently, silently building. These questions grow even tender when we consider something called germline editing. This is when CRISPR is used not on a grown person, but on an embryo, before life has truly begun. An edit made here is permanent. It doesn't just affect the child, it affects every child that child may have, and every child after that. It echoes down generations. A single choice ripples forward into futures we cannot yet see. Many countries have placed restrictions on germline editing. Some have banned it altogether. Not because they don't believe in science, but because they believe in pacing it with wisdom. They are not saying no. They are saying not yet. Because when we move too fast, we may carry forward our biases, our assumptions, our mistakes. We may decide what is normal too narrowly. We may begin to erase not illness, but difference. And in doing so, we may lose something sacred. The world is full of people who are born different, not broken, just unique. People who see and feel and move through life in ways we don't always understand. To edit them out is to forget that diversity is not a flaw. It is nature's most powerful design. Still, the questions remain, and they are not meant to paralyze, but to guide. There is hope here, not just for healing, but for humility. Because CRISPR also asks us to look inward. What kind of world do we want? A world where we erase every mistake, or a world where we learn to live more kindly with imperfection? Can we create without controlling? Can we innovate without excluding? Can we change the code and still remember what it means to be human? These are not questions science can answer alone. They are questions for all of us, the curious, the cautious, the caring. And while the future unfolds, science listens. There are committees now, ethical reviews, open debates, global conversations. Many scientists have pledged not to move forward with certain uses of CRISPR until the world agrees, or at least until more voices are heard. And this is what gives the story its shape. Not just discovery, but reflection. Not just possibility, but purpose. You, lying here in the hush of night, are part of this story too. Because the future is not written by machines. It is written by hands, by hearts, by choices made in quiet moments. Moments like this, where a question lives longer than an answer, where pause becomes wisdom, where silence means we are listening. So let this stillness hold you. Let the questions echo, not to disturb, but to remind you of something tender. Science is not just what we know. It is what we choose to do with what we know. And tomorrow, or the day after, someone will sit down with a strand of DNA, with a sequence of letters, and ask not just what they can do, but why. And this why will shape everything that comes next.
You are resting now. Somewhere between wakefulness and dreaming. That quiet space where imagination softens, where questions float like dandelion seeds in the dark. And so here, gently, we look forward, not to predict, but to wonder. What might life become if we carry CRISPR not like a sword, but like a lantern? Imagine a world where disease is not battled, but prevented, not with fear, but with foresight, where a child born with the threat of inherited blindness is offered not a lifetime of darkness, but a simple injection that restores the light. This is not fiction. This is already beginning. In trials around the world, scientists are using CRISPR to edit genes in the retina, the delicate back layer of the eye where sight begins. They are correcting a mutation, and slowly sight is returning. Not instantly, not perfectly, but like dawn seeping through fog. Imagine the grace of that. A future where light is not lost, but remembered. Now drift with that thought to the blood, where scientists are using CRISPR to re-engineer immune cells. They are training these cells, like shepherds guiding flocks, to recognize invaders more clearly, to find cancer where it hides, to fight back not with poison, but with clarity. And the research continues to edit the liver, the lungs, the brain, to one day treat Huntington's, ALS, Alzheimer's, not by easing symptoms, but by changing the script that wrote them. The goal is not to escape mortality. It is to ease suffering, to offer time, not eternity. But this is not only about the sick. CRISPR may touch the healthy too. Picture a world where a simple swab of your cheek gives doctors a map of your risks, your chances of certain cancers, or heart conditions, or rare reactions to common medicines.
And with that knowledge, your treatment becomes not general, but personal. This is called precision medicine, a way of caring where the path is shaped to your body, your code, your needs. CRISPR is one of the tools that makes this possible. But what if we looked beyond our own bodies? CRISPR may shape the food we eat, not by adding mystery, but by restoring balance. Imagine crops that can grow without pesticides, that resist drought, that nourish more deeply. Bananas that don't rot too quickly. Wheat that doesn't trigger illness. Trees that absorb more carbon from the air. Imagine helping the earth not by taking more, but by editing less waste, less harm. In labs, scientists are using CRISPR to alter the genes of mosquitoes, to slow the spread of malaria. In time, perhaps entire populations of disease-carrying insects will be transformed, not killed, but changed in ways that break the cycle of transmission. These are careful steps. Nothing is rushed. Every change is tested again and again, like a whisper, repeated until it becomes a song. But even as we marvel at this future, we return to humility, because the more power we hold, the more patience we need. CRISPR does not promise perfection. It does not erase hardship. It offers something subtler: choice, direction, possibility. And with it comes the responsibility to listen to the science, to the ethics, to each other. We are not writing a new species. We are learning how to tend to the one we already are. You, lying here now, are part of this moment in time. A moment when humanity reached deep inside the molecule of life and found not chaos, but a pattern. Not a wall, but a doorway. The door is open, but we are not running through. We are stepping forward slowly, hand in hand with caution. In schools, students will one day learn about DNA not as a mystery, but as a medium, something to be read, respected, maybe gently revised. In hospitals, gene therapy may become as common as antibiotics once were. In homes, families may speak of genetics not as destiny, but as dialogue, a conversation between what is given and what is possible. And in all of this, there will still be love, still be art, still be dreams that have nothing to do with genes, because we are not just biology. We are stories. We are spirit. We are stardust wrapped in skin. CRISPR may guide how cells divide, but it cannot tell us how to be kind. That part is still ours. Let that be your lullaby tonight. Science can shape what we are, but only we can shape who we become. You have journeyed from the spiral staircase of DNA, through the whispers of bacteria, into the hands of scientists and the questions of ethicists. Now you rest at the edge of what comes next. You don't need to solve it all tonight. You don't need to understand every term, every technique, every trial. You only need to remember that this future, written not in fear, but in light, is unfolding in real time, like a blossom opening toward the sky. And as you fall gently into sleep, let your breath become the rhythm of wonder, the rhythm of science, the rhythm of life still being written. Because even now, as the world spins quietly beneath the stars, somewhere in a small lab by a soft blue light, another base pair shifts, another letter is rewritten, another possibility begins.